Scan for Sovi, your AI bestie. I got your back.
Prokaryotic gene regulation can be tricky, but we can solve this attenuation question fast. Let's look at how transcription and translation work together right now.
Attenuation relies on the ribosome moving along the mRNA while it is still being built by RNA polymerase. This coupling of transcription and translation is the key to the whole process.
In prokaryotes, there is no nuclear envelope to separate these two events. This allows ribosomes to jump onto the transcript immediately, which is exactly what makes this specific mechanism possible here.
Eukaryotes separate transcription in the nucleus and translation in the cytoplasm. Since they are separated, option B is the only logical answer. Well done! Scan the code to learn with Sovi. Watch signs and avoid rush. Trust given info and check steps for option B.